have to really move on with the session. Okay, I hope it's fine. And let's start. Okay. So what did we study in the last class? In the last class, I think I talked about, the termination, right? So we finished the whole transcription, but we have to talk about the post-transcriptional machinery. So what did we learn in the transcription? The most importantly, what we learned is the termination. Correct, no? So like obviously we learned about the promoters. We learned about what is 10 regions, what is 30 regions, and what is RNA polymerase. So why RNA polymerase is a holoenzyme, and what is the energy sequence, energy for this particular elongation of mRNA. So what is the role of each and every protein? We discussed in so much detail in the last class, correct, right? So now, since we have finished till the termination, in the termination also we understood that it is row dependent and row independent termination that happens generally. So now we have an idea like how the general transcription takes place, correct, right? Now today's class, what will happen is now we have to go and understand something called as post-transcriptional machinery. Now, what is post-transcriptional machinery? If you look at the nature of the nature of RNA is quite catalytic and unstable at the both on the same time. What is catalytic? It is very, very abrupt and it has a very short lifetime. Okay. So RNA is very, very important information you can understand, right? So that is why RNA has to be protected and also DNA. DNA should be protected. Now, what happens? You know that we have two types of cells. One is your prokaryotic cell, where the nucleus is just floating in the cytoplasm without any nuclear membrane. And then you have your eukaryotic cell. And in eukaryotic cell, you can clearly see that there is a clear distinguished nuclear membrane. And within the nuclear membrane is a very well organized genetic material in the form of DNA or chromosomes. So what happens, guys, here, the the rate of transcription. See, for example, what happens? What is the role? DNA should become what? RNA, not become, like DNA is read into RNA, and RNA becomes what? Finally, it is protein. Correct? No. So we know that this is a central dogma. So the process from DNA to RNA is called as transcription. Yeah, yeah.
Good evening, sir.
Yeah, very good evening. Yeah, like so the process of conversion of DNA to RNA is called as transcription, and from RNA to protein is called as translation. Correct, right? So this process is very, very abrupt, means very fast. So when it is very, very fast, so now the control on the gene becomes very, very less. Correct, right? If you, for example, let's say that the RNA says produce around 10,000 units of particular protein. But the body does not need 10,000 units of protein. It requires only 200 or 300 units of protein. Now, how can they stop this process? Stopping this process becomes very, very difficult. You understand my point? So the machine is not well evolved, I'm telling. Simple. In simple terms, the machine is not well evolved. But if you see in eukaryotes, our DNA cannot be tampered so easily. Tampering means no hijack. You cannot hijack our DNA so easily. So that does not happen basically. So here what happens? We are protected by a nuclear membrane. So there is a proper communication way, okay, in which what comes in and what goes out of the nuclear membrane, especially the genetic material. So even RNA is a genetic material, right? So an unknown RNA coming inside, an unknown DNA sequence coming inside or going out, this is quite prohibited, or it is not allowed, or it does not allow because of the presence of nuclear membrane. You get my point? So this is first reason. And what is the second reason? The nature of your mRNA. So what is the nature of the mRNA? If mRNA is not modified, that means after the production in eukaryotes, what happens, guys? So we have the DNA. When the DNA is translated, okay, transcribed, okay. After transcribed, what does it become? It is not matured mRNA. It is called as heteronuclear mRNA. Okay, it is called as, it's actually small letters. Okay, HNRNA. So they are called as heteronuclear RNA. So the reason is there. What is the reason? First thing, the heteronuclear mRNA, let's say that, okay? So let's say that the DNA is being transcribed and we have a hnRNA. Now, if the hnRNA is not modified, let's say if it is not modified, it can last only 30 seconds. Maximum 30 seconds to 60 seconds is the maximum lifespan of that particular hnRNA. What benefit does it get with the post-transcriptional machinery? With the post-transcriptional machinery, the lifespan of the hnRNA is extended from 30 seconds to 60 seconds all the way till 45 minutes. Look at the exaggerating difference in time, okay, just because of some modifications that happen in your mRNA. So it gives extra stability to the mRNA. First thing, post-transcriptional machinery, what does it do? It gives the mRNA exceptional stability so that it can live in the cell for a longer period of time. Since there is a nuclear membrane, it helps in the transportation of the mRNA from the nuclear region all the way to the cytoplasm. So that is the second thing. And what is the third thing that happens? The gene that is being transcribed is not completely functional. That means what? If you look at an eukaryotic gene, the eukaryotic gene, which is a DNA, is not continuous. Okay, it's not continuous. What is the meaning of continuous? If I start from reading my gene from first base pair all the way till the last base pair, do all the base pairs make some sense? That is the question mark. Here, it does not make sense. So why it does not make sense? If I look at my eukaryotic DNA, it is interrupted by something called as it is interrupted by sequences. Let's see here. See, this is the eukaryotic gene. Here it is divided into, you can understand that the gene has two segments. What is that? One is called as an intron sequence. Other one is called as exon sequence. Sorry, I marked exon. Yeah, exon sequence and intron sequences are present. What are these sequences? Already in genes, you can see that exon means what? Express. Expressing sequence. What is intron? Intron means interrupting. Right? So we know the understanding of interrupt. You know the meaning of express. Now I am expressing, guys. Correct? No. Now I am expressing. So this is my expression. Now what is interruption? Interruption is something which is not related to the class. When you talk something related, unrelated to the class, yeah, I'm talking. I'm going to repeat. Okay. So what is? See, I was trying to tell that we have genes. What genes? Eukaryotic genes. As I was telling, if you take eukaryotic genes, that is the genes then which is in our DNA, so which we are not continuous, we don't have the continuous reading frame. Continuous reading frame means what? If I start from the first base pair all the till the last base pair, I can read, and obviously even the nonsense sequences I can read, but it does not make any sense. You get my point? So what is happening here? My sequences are interrupted. What sequences? See, if I look at an eukaryotic gene, I can see that there are two types of sequences. One is called as exon sequences. Other one is called as intron sequences. What is exon type of sequences? Exon means express. Okay, exon means what? Expressing sequences. So expressing sequences means what? Something that makes sense. Something that results in the formation of protein. That is called as expressing sequences. What is intron sequences? Intron sequences are called as interrupting sequences. Okay, so these sequences are called as nonsense sequences. Okay, nonsense sequences or interrupting sequences, so they are called as that. So now, why do eukaryotes have intron sequences? Okay, sir, repeat from beginning of the transcription. Oh God, that will be a difficult task. No, I will see. I cannot promise to post-transcriptional machinery. Yeah, that's what this is the beginning of the post-transcriptional machinery, basically. So, okay, I will summarize for you. What we are talking about is when you talk about post-transcriptional machinery, what is post-transcriptional machinery? So basically, editing of mRNA is called as post-transcriptional machinery. Why editing has to be done in the mRNA? I gave you two reasons. One is we are an eukaryotic cell. That is the first reason. And the second thing is the nature of the mRNA. First reason we talk about why eukaryotic, being a eukaryotic cell, you need something called as post-transcriptional machinery. See, our eukaryotic cell is very well guarded. So that means our nucleus and nuclear membrane, they play a very important role in separating things. They do not allow anything to enter or anything to exit without the permission. You understand my point? So for your mRNA that is being produced in the nucleus has to exit into the cytoplasm. So to this exit, it has to pass through what? Nuclear membrane. Does the nuclear membrane simply allow your mRNA to go through? Definitely no. For that, what do you need? You need modification. Simple. You need what? Modification. This is the first reason. Second thing is what? The nature of mRNA. So when I'm talking about nature of mRNA, basically the unedited mRNA. What is unedited mRNA? Our HN-mRNA. Our HN-mRNA's lifespan is only 30 seconds. Less than 30 seconds, basically, it will get denatured very, very fastly. So now with this editing, what will it give? With this editing, its lifespan will get expanded from 30 seconds to all the way till 45 minutes. Okay. Now what happens in this editing? So basically, HNRNA is converted into a mature RNA. Okay, it is converted into a mature RNA where it is stabilized and it is made into a continuous expressing RNA. Okay, so now stabilizing, I will talk about it. The second part, what I told, the continuous expressing part. So that is what we were talking about. Now you understood why this post-transcriptional machinery will have to take place. Now I was talking about the nature of mRNA. So this is the beginning. Now the topic is starting. Which one? What happens in post-transcriptional machinery? Where is post-transcriptional machinery is happening? We know that it is happening onto the RNA. What RNA? mRNA. Is it a proper mRNA? No. What type of mRNA it is? It is an HNRNA. Where is it found? It is found in eukaryotic cell. Okay. Now first we have to understand what type of RNA it is. If I look at a prokaryote, the mRNA is having a continuous reading frame. That means from base pair 1 all the way till the last base pair, all of them will result in the formation of what? Protein. Okay. But our eukaryotic RNA is not like this. Our eukaryotic RNA is quite interrupting. Okay. How it is interrupted? See, if this is the DNA of a eukaryote, the DNA of a eukaryote consists of what? For example, this is expressing, this is non-expressing. This is expressing, this is non-expressing. Okay. Now, DNA becomes what? RNA. So, even if you look at our RNA, it has two sequences. The RNA transcript will have two sequences, two types of sequences. One is expressing sequences, other one is called as interrupting sequences. Hold on, guys. HNRNA, heteronuclear RNA. Okay. Sir, please little bit slow down. Yeah, guys, because I'm repeating for the third time. That's why I'm rushing. Okay. Yes. I will slow down. I will slow down from here. Okay. Sorry, guys. Because it's repetition, so I thought it would be quick. Already 20 minutes gone. So sorry about it. Now I'll go Shanti. Okay. So what I'm saying is, if you look at your RNA, you can see that the RNA transcript will have two sequences. So one is expressing one, other one is called as interrupting one. So this interrupting is there. No, interrupting is bad, correct, right? Interrupting is bad. Why interrupting is bad? Because it does not make any sense. It is not giving rise to any protein. So now, what are these interrupting sequences? We will see. See, since we are eukaryotes, there are two arguments to it. Okay. What is the two arguments? Some say that it is junk. Junk means what, guys? Waste. Some say that it is ancestral. Okay. Now, what happens? What is ancestral? My spellings, forgive me. Okay. Some scientists say that, see, some waste DNA started accumulating over the period of time, and those are called as introns. So scientists could not give any proper reason to it. You understand my point? So those are called as introns. Some scientists, what they said, see, these introns are not waste all the time. So as we evolved through, as the evolution took place, some of the characteristics went under modifications. No, we have gone under so many modifications over the period of time, right? From several millions of years. So this modified characteristics which we did not want, which we don't want anymore. So those have converted into an intron sequence. So that is what others people will tell. You get my point? So some others will also argue that since the gene expression is so complex and so sensitive, we need to have control on the whole system. No. So to gain the control on the whole system, we need introns. Why? So that we can delay the process of transcription to translation. You understand my point? So the three reasons are given why the introns are present. Okay. Oh. Okay, why the introns are present. You understand my point? So what are interrupting sequence? I told you it is nonsense sequence. Sometimes they tell that it is ancestral DNA. Some people will tell that it gives control over the transcription and translation. So many reasons are there why the introns are there. But having an intron in the mRNA will make mRNA non-functional. That means it is not useful. So we need to make this mRNA quite useful. Okay. Yeah. I believe that introns are the cause for the ancestral characteristics. Yeah, we have gone under so many modifications. So there is a high chance that these introns are the remnants of the changes that happened for a particular gene. Correct? No. For example, you can even talk about the hair. We lost the fur and we became bifids, and so many characters have changed. No. So I think it's an ancestral. Not me. Many scientists argue, and they it's very well backed. Which one? Introns are ancestral gene. Okay, so that is the one thing. What about exon? Exon is expressing what I will tell. I'm telling you what is exon. Okay, now we understood what is intron. No, intron is unnecessary. So with intron in the mRNA is not mRNA. It is basically hnRNA, which is called as heteronuclear RNA, which cannot be functional. When I say cannot be functional means directly you can't synthesize a protein. Okay. So directly you cannot synthesize a protein. That is why what happens, you need to remove these introns and stitch all these exons together. Okay. So when you stitch all these exons together, your RNA transcript becomes continuously reading. Okay. You can see that the RNA becomes continuously exon. So all these exons together will form one proper functional mRNA. Did you understand, guys? So we understood what is the nature of the genes that is present in the eukaryote. So I told that eukaryotic gene is not continuous, it is interruptive. So they have interruptive sequence in the form of introns. So when the gene is transcribed, the hnRNA contains expressing sequence as well as interrupting sequences. So we have to remove interrupting sequences and stitch all the expressing sequences to make our mRNA functional. Okay, this is what I do. So now we have understood the nature of the mRNA. So now the next part is we have to see what does our post-transcriptional machinery include. Our post-transcriptional machinery includes three important steps. Okay, it includes three important steps. What are those? One is called as capping, and the second one is called as tailing. Okay, and the third one is called as splicing. I will see that out 1 second. Okay, so capping, tailing, and splicing. Okay, I'll tell you why it is exon 1, exon 2. Okay, give me a few seconds. You will understand as we move further. Okay, so capping, tailing, and splicing are the three important steps that will take in the post-transcriptional machinery. All three are very, very important. Okay, so let's see what is capping. So now, if you look at capping, now what will happen? I need some space. I will use this. Okay, so now in the capping, you have the 5' end and you have the 3' end for an mRNA, right? So at the 5' end, a special cap is added. What is that cap? It is called as 7-methyl gynosin cap, or it is also called as 7-met cap. It is also called as 7-met cap, or it is also called as 7-methyl gynosin cap. So now what is happening? This is your RNA at the 5' end. A cap gets added here, and this cap is called as 7-met cap. Okay, and the same phosphodiester linkage will happen with the 7-methyl gynosin cap. Okay, a phosphodiester linkage will establish here. Now, why do we require this cap is the first question. Now we require this cap because of two reasons, one and two. The first reason is it adds stability. Okay, it adds stability to the RNA. Very easy. What is the second reason? You see, once the mRNA comes out of the nuclear membrane, who has to receive this mRNA? Your ribosomes, correct? No. So ribosomes, what should happen? Ribosomes should recognize this mRNA and should accept it. Correct? No. So this 7-methyl gynosine cap that is present here, what it will do, it will give a space or a platform for the translation machinery to start. Okay. For the translation means what? Your RNA should become protein, no? So for that process is called as translation. The translational machinery will come and assemble at the 7-methyl gynosine cap. So that becomes like a start sequence. That becomes like what you tell a promoter region that we studied. Similar to that, it becomes that recognition sequence or recognition factor for your ribosomes to accept. Okay, this is my mRNA. So I will go assemble here and start synthesizing the protein. So first one is it adds stability. Second one is it gives a facility for the translational machinery to come and assemble. So this is done. So that is called as capping. Next one is tailing. First we'll go for tailing. What is tailing? Tailing means you have the 5' end. In the 5' end, we have added a cap. And then we have a 3' end. In the 3' end, what happens is a poly A tail. Okay. A special enzyme called as poly A polymerase, a type of RNA polymerase. Okay, a special type of RNA polymerase called as poly A polymerase. What it will do is it will come at the 3' end. Okay, it will come at the 3' end. At the 3' end, what it will do is it will start adding 150 base pairs to 200 base pairs long adenine, 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 adenine tail. So there will be a long adenine tail within your mRNA. At the 3' end, it will add. Now, why this tail got added? Why there is a need for adding an adenine sequence? Simply, the reason is first one, again, stability. Okay, stability. What is the second reason? Second reason is, see, this is your mRNA. Okay, not exactly, but imagine that this is the mRNA. This is the 3' end. And here, what do we have? We have a poly A tail. Correct, no, guys? Now, imagine that this is the nuclear membrane. This is the nuclear pore. Okay, this is called as what? Pore. And this is the nuclear membrane. Okay, I'll write it as NM, nuclear membrane. Now, see here, this poly A tail will go and attach to the gates of the nuclear membrane. It will go and link to it. When it links to it, the port or the pore of the nuclear membrane, which was closed, or the gates of the nuclear membrane will open. Now, what will happen? The 5' end will get sucked in and the 3' end will get attached here. You understand? So now what will happen? The 5' end will go out and followed by the 3' end. What happened here? With the addition of a poly A tail, okay, now it helped the RNA to move from the nucleus to the cytoplasm. Simple. What is the second reason? Transport. Sorry. What is the second reason? Transport. Transport is the second reason. Got it? No? So the poly A tail is added for the transportation. So that is the second. Now let us go for the splicing. Now we understood the capping, we understood tailing. Now our thing is we have to go for splicing. Now what is splicing? Spliceosomes are special enzymes. Do I have a space here? I will try to use the space, guys. Okay, don't mind the heading. So spliceosomes are the enzymes. Now, the spliceosomes take help of snRNA. What is snRNA? snRNA is called a small nuclear RNA. So, small nuclear RNA, we will get 5S rRNA is another one example of snRNA or small nuclear RNA. Now, what this 5S rRNA will do is, now we have, sorry guys, we have introns, exons, introns, and exons. So we have sequences, no? This 5S RNA will go and bind to the spliceosome, and it will help in recognition of what your intron sequence is. Okay. So the spliceosome, what it will do, it will act like an endonuclease. The spliceosome, what it will do, it will act like endonuclease. Last class, I told you what is an endonuclease. Okay. It will act like an endonuclease. Now, what will happen with the help of 5S RNA? The spliceosome will cut, for example, if this is your gene and this is the intron, this end and this end will be removed. This is exon and this is exon. Okay, this is intron, let's assume. Now, what will happen? This will come like a bubble. You know, this will be pulled, and when you pull this, what will happen? This thing will fold like this, correct? No? I hope I'm trying to explain. Let me write it. Something like this. Now, this is your intron. This is, this is your exon. This is your exon. This is your intron. Now, this will be like bubbled out and two exons will come and attach with each other. Now, instead of intron, exon, what do we have? We have exon, exon, which is a continuous reading frame or continuous expressing sequence. Now, what will happen with capping, tailing, and splicing? You have a matured mRNA. Okay, this mechanism is called as post-transcriptional machinery. Did you guys understand? Yes, no? I'll ask you true or false. You just tell me if you understood or not. Yes. Yeah. Perfect. Cool, guys. Chalo. Can we go for the next one? Now you see the when you look at the whole transcriptional process, the transcription do not involve only making one type of RNA, guys. So we know that it involves manufacturing of three different types of RNA. Correct, no? So when does mRNA transcription take place? When does tRNA transcription take place? When does rRNA transcription takes place? All of them happen simultaneously, you see. So if a gene is transcribed for the gene, a respective rRNA, a respective tRNA, a respective mRNA is transcribed simultaneously. Now, why do we require three different types of RNAs? Now we will see. Why do we require rRNAs? Now, if you look at rRNA, it is also called as ribosomal RNA. So ribosomal RNA, what it will do is, it helps the ribosomes to recognize mRNA. So that means what? When the mRNA is manufactured or it is produced, the role of mRNA was to make sure that the ribosomes understand what protein to synthesize. Okay, they have to understand what should I manufacture. So that message is where it is present in the mRNA. So how will the ribosomes understand that I have to start the process now? So that is facilitated by your rRNA. So what rRNA will do? First, rRNA will go and bind to the ribosome. Now that will lead to the activation of ribosome. What is activation of ribosome, guys? Now you know that ribosome has what? Two subunits, correct? No. It has two subunits. A large subunit and then you have small subunit, which are actually separated. They're basically separated. Now, what happens when the rRNA comes, to play, when it goes and binds the small subunit? Now, immediately ribosome gets activated. Now, they will go and assemble onto the mRNA and start the protein synthesis. Okay. So that is the one thing. So it also acts as an enzyme, ribozyme. Okay. So which activates the ribosomes. And finally, the role of ribozyme is to produce polypeptide bond. Very, very important. The polypeptide bond or the peptide bond synthesis happens because of the rRNA. Clear, no, guys? So this is the functions of rRNA. Okay. So next one. If you see, there's a lot of information here you can go through, but we'll stick to what is significant here. Okay. Next one. Next one is the why tRNA. See, rRNA though we understood that that if we have the rRNA, then it will lead to the activation of ribosomes, and then it will go for the protein synthesis. Now, there is one more RNA, which is the tRNA. Now, if you look at the protein synthesis, what is your proteins? If I ask you what is your protein made up of, TI. Okay, what is the protein made up of? What do you tell? You tell that it is made up of peptide chains. Peptide, okay. Peptide chains, okay. Now, what is this peptide chains made up of? See, a protein is a polymer, simple. Okay, it is made up of several types of monomers. So these monomers are called as AA, or it is also called as what? Amino acids. Correct now, guys? So now what should happen for a very particular protein to be synthesized? A proper sequence of amino acids have to come in place. You know, one amino acid after another, another amino acid after another. So a proper arrangement of amino acids have to take place for a construction or for the synthesis of a polypeptide chain or a protein. Now, who brings these amino acids sequentially, or who brings the amino acids for the synthesis of your protein? The answer is the tRNA. Okay. So now what happens? The tRNA is actually the sole member who is responsible for bringing the amino acid. Got it? No? Now, what is tRNA? The tRNA almost consists of 10 to 15% of the total RNA in the cell. That means you see they are very, very large in volume. Okay. So now, if you look at the tRNA shape, it looks like something called as claw leaf shaped structure. Okay. It looks like the claw leaf shaped structure. So the structure of tRNA was basically suggested by Robert Hawley in 1965. So basically, he got a Nobel Prize because of this particular what do you call description of the tRNA structure. Now, guys, this remember this. This diagram is super important, very, very important. You can go through many previous year papers, module papers. You always come across some or the other way. You come across this particular diagram at least some alternative times. So what we have to study from here? The labeling. Labeling is very, very important. So first we will see the labels and we will see the functions of what these particular labels mean. Okay. Now, since it is a tRNA, does it have 5' and 3' end? Yes. So we have a 5' end and we have a 3' end. Okay. So the 5' end is called as acceptor step. So we can also call it as amino acid acceptor end. Okay. Amino acid, where the amino acid comes and binds. No. So that is called as a 5'. Got it? No. See, along with the 5' end, what do you have? You can see that there is a D arm. And after D arm comes what? You have an anticodon loop. Okay. And then after anticodon loop, what do you have? You have a variable arm. Okay. Which is also called as T-Ci-C loop. And then what do you have? You have a T arm. And then you have a long 3' base. So you see here, the long end is a 3' and the short end is the 5'. Okay. I think I should have a different diagram. No, it's gone. Anyways, guys. So you remember this. The longer end is called as 3' end and the shorter end is called as a 5'. Got it? No. So this is very, very important. So what do we have here? At the 5' end, it is called as amino acid binding site, or it is also called as amino acid acceptor arm. So this is called as amino acid acceptor arm. Next one, what do you have? We have DHU arm, or it is also called as a D arm. And then we have anticodon loop. And then we have T-Ci-C loop. And then we have T arm. And then we have the 3'. Got it? So now each of these have a specific function. So let's see what is this specific function. Okay. Now, if you see the T-Ci-C arm, okay, so that is the variable, the T-Ci-C arm. What is the function of this? So they basically help the ribosome to bind at the time of protein synthesis. As simple as that. Okay. Now we have the D arm. What is the role of the D arm? So let's see here. The DHU arm, what does it do? So they help in recognizing specific amino acid sequences. Okay. So they help in recognizing specific amino acid sequence. So this one will help in, binding of ribosomes. Let me write it here. So this one, what it will do? Recognition of amino acid sequence. Okay. And it also helps in activation of amino acyl synthetase. Here it is, guys. Amino acyl tRNA synthetase. So this is an enzyme. Okay. So this particular arm is responsible for bringing particular amino acid. So if I want a particular amino acid, this arm is the one that helps in recognition. What is anticodon loop? What does it anticodon loop contains? It contains 7 unpaired nitrogenous bases. Okay. So they are which are quite complementary. So what is the function? Basically, they carry the amino acid towards the site of protein synthesis. So that is the whole function of your tRNA. So guys, remember, more than the functions, you have to make sure that you understand the cloverleaf structure of tRNA and its labeling. Again, 5 prime, D arm, anticodon, anticodon loop, anticodon arm, which is also called as T-C arm, variable arm, T arm, and then you have your 3 prime. Please go through this diagram as many times as possible and make sure that you get too familiar with it. Okay. So now the tRNA. Before understanding your translation, we have to understand the translation, right? So translation means you have the RNA, we have the DNA. DNA became RNA. We understood the process where through the transcription. After we have the messenger RNA or the functional mRNA. Now eventually this functional mRNA transcript becomes what? Protein. So that is a whole gist of it. So now we have to understand what is a gene and how the genes are actually arranged and what does gene contains and everything. Okay, so all these things. So let's see. We see later on what is this and yeah. So basically, our gene contains something called as codes. Okay, so the prior slide was also important. Um, yeah, I don't mind. Let me explain this. This is also very important, guys. Sorry about it. I thought of skipping it, but I'm not feeling to because this is very important for examination. And I'm also lacking very less time, okay, to finish this lesson. Anyways, so let's see. What is the gene? Basically, in 1857, our Mendel started experiments and he called them as factors. And in 1909, we have the Danish botanist, which is called as, Janssen, and he proposed that something there is a chromosomes and it controls the phenotypes. Okay, and he gives the term called as genes. And then we have Beadle and Edward. So you don't have to remember this. So it's a journey. So they propose something called as one gene, one enzyme hypothesis. So each enzyme, each gene is responsible for each protein or each enzyme. So they proposed it. And then we have Oswald and Avery. So they told that they are very what do you tell? The DNA is the genetic material, and then you have Watson and Crick. They suggested the structure of DNA. So basically, when you look at a gene, this is an important part. If you look at a gene, they are made up of specific sequence of DNA nucleotides. When I tell you specific sequence means the arrangement is very well pre-thought. I don't know, like evolution has thought about it or God has designed it. Okay, so that is it. So there is a specific, very highly specific sequence of this DNA nucleotides. Okay, now we have our Benzer. So the Benzer, what he did is he proposed a model of concept of gene. Okay, while he was working on E. coli. So according to him, what he told is a gene can be. This is a very important. A gene can be a cistron, a recon, a muton, and a replicon. Remember this. A gene can be, for according to him, what is a gene? A gene is a cistron, a gene is a recon, a gene is a muton, or it is a replicon. Let's see what is the meaning of each of these things. What does a cistron means? It is nothing but a structural gene. Okay. So a cistron is you have a promoter sequence and followed by a gene. Okay. So which is a functional unit of DNA and it codes for a specific protein. This is a protein coding sequence. I will call it as this is a promoter region and this is your structural gene. So this is controlled by a promoter. No. So now sometimes what happens in this cistron, if you have one promoter, and let's say this is gene 1, this is gene 2, this is gene 3. So this is G1, G2, I will write G1, G2, G3. So now you see that there are more than one cistron, correct? No? So this will become you can divide the whole genes into polycistronic as well as monocistronic. Okay. Now, if I have one promoter controlling several genes, it is called as monocistronic. Sorry. And if I have one promoter, one gene, that is called as polycistronic. So something like this. You get my point. So that is called as cistron. What is a recon? So the unit of DNA that allows changes. You can see that even though we have the same human-like characteristics like our parents, but we don't appear the same. That non-parental characteristics is there, no? So that is existing, that comes into existence because of a particular DNA that allows recombination. Okay, that is called as recon. What is a muton? Any part of a DNA that has undergone or it is capable of undergoing mutation. So that is called as a muton. Do we have genes that act as a muton? So there are particular genes. For example, if you look at E. coli, and E. coli you have beta galactosidase gene. Correct? No? If you see in future classes, you can understand that beta galactosidase is almost like a mutagenic gene. Okay. So it is a muton. So this particular DNA undergoes or it is capable of going under mutation. So this is called as muton. What is replicon? So it's not replication, it is replication. Any unit of DNA that can divide itself and make copies of itself, that is called as replicon. So what are the different ways a gene can exist? It can exist as a cistron, a recon, a muton, as well as a replicon. According to who? According to Benzer. Remember his name. Very, very important. Benzer suggested these modules. He is the one who told that a gene can be in any of this form. It can be a cistron, a functional unit of gene. It can be a recon, a gene that undergoes recombination. A muton, a gene that can undergo mutation. And a replicon, a gene that can replicate. So these are the forms of DNA that we can see in an organism. So not DNA, I can just call it as genes or factors that the Mendel referred to. So that is how the gene can exhibit itself. Got it now? Excuse me. Guys, are you following me? Sorry, guys. Yeah, sorry for the inconvenience. It's the sneezing. Yeah. So let's go for the next one. Okay, genetic code. It's beautiful to understand genetic code. It's very easy concept. Too. It's very easy. You can understand. I know you guys are smart, and you can pick up the concept very easily if you just give the attention. But you just have to imagine how perfect it is, guys. How perfect it is. If you see from an amoeba, a bacteria, or a non-chordate, a chordate, a plant, any cell you take, and if a particular, let's say that if this is AUG, is a code, and if it has to code for a particular amino acid, okay, what is amino acid AUG code for? It is methionine. Okay, met. Well, let's call it as met. So AUG codes for methionine not just in bacteria, in human beings, in dog, in cat, in lion, in tiger. If the AUG comes in your DNA, it codes for methionine. So the universality of these codes is something that is fascinating. That means it gives us a very clear picture that we are not much different. Chemically, we are all same. We have all evolved through the same roots. We have come from the same tree. So that is what something this particular topic gives us. So what is a genetic code? So basically, back in the days, the Corona, okay, his name is Corona, okay, and a very noble scientist. When he was working in the laboratory, he wanted to see how the genetic codes, a lot of experiments, artificial synthesis of genes have been taking place. So now, as the experiments proceeded, the first code was done for uracil, okay, a polyutane was UUU sequences were what do you tell, or synthesize in the lab. So as they synthesize in the lab artificial mRNA, then they synthesize amino acid sequence out of it. So they have found out that out of, let's say that we have 99 sequences of your uh, this is a 99 sequence, okay, 99 numbers of you are there. So when they found out how many amino acid sequence were there, they were like 33. So something hit them that okay, each code contains of three letters. Each code should contain of three letters, and these three letters code for a specific amino acid. So that is what they came across. That is the conclusion they first thought. So if you look at our code, so what is a code? Our code is nothing but a three, okay, specific nucleotide sequence. So what is a specific nucleotide sequence? For example, let me write a random genetic code. Okay, so this is a 5' end, guys. I'll write it as A, C, 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 G, A, T, T, G, G, C, C, A, G, G. Okay, this is my sequence. Got it. So this is my DNA. What will be my RNA? A will become U, and G, C, U, A, C, C, G, U, and then you have your CC. Correct, no, guys? So this is my RNA. Now, RNA becomes protein. No? RNA is read as protein. No? How is RNA read as protein? Now we divide this into three. 1, 2, 3, 4. Okay, 5. Sorry, 5. And then we have 6. Not bad. Not bad. I finished the code. So now you see here, this is equal to one amino acid. This will become another amino acid. This will become another amino, not become, this will read for another amino acid. U means this one specific amino acid. For example, you can see here, where is our U? Can you find U? Uh, yeah, U. What do you have? We have phenylalanine. You can see here. U is what? Phenylalanine. Like that. You can see UUC, phenylalanine, UUA, leucine, like that. CUU, it is also called as leucine. Here, isoleucine, valine, like this. Are you following me, guys? So, like this, three letters will make up to one code, and each code will code for a specific amino acid. Okay. And then what do you have? So we have codes and anticodes, and anticodes are something we have learned during the transcription. Correct? No? Or yeah, we have 3 minutes. Can we wind it up here? We'll take up in the next class. Okay? Because half of it is not good, guys. I don't like it. So, as usual, some people are okay, some people are not okay. But I would still like to get your feedback. And I have been telling you guys, please, whoever has the problem with the class, and if you're not understanding a specific topic, you can be free and you can ask me. Okay, I have all the patience to repeat, and I enjoy repeating and telling you guys. Uh, you should ask me. That's it. Okay, so until and unless you don't open up, I can't understand. No, today is very bad, or the class was so bad. It was that bad, students. So, no, anyways, I accept it, and let's see what we can do. I don't understand where is the problem. Alphabetic is language of humans, but how can our body know it? We are not having alphabets. Basically, we are having a sequence of nucleotides. It's just molecules. If you see, these are like you understand what is a nucleotide, right? So now these nucleotide sequences are arranged as adenine, thymine, cytosine, guanine, like that. They are arranged in the DNA. So we are giving the alphabetic form to understand the codes. So that is very strange, how these codes can mean so much and it can be so definitive. You see how defining it is. It does not make any mistakes, and it changes. It gives allows so many things and everything. I will give my email ID. So, so this is a miracle. I can't say why. There are so many things I can tell you. How the universe is not just physical universe; it's numericals. Correct? No, you can't define the universe with what do you tell? uh, with size. You can tell it with codes like numbers and time. So similarly, even your body, it's not something like too much, guys. It's creation. You can just appreciate the creation. That's it. It's simple. Okay. But anyways, we have the capacity to understand what has happened in the evolution. So yeah. Anyways, good question. Sorry if I dragged your answer. Yeah, it's been my question since long back. Anyways. Okay, guys, thank you. I've dropped my email ID who were asked. Okay. Guys, I don't really like, of course. Yeah, really like to see you not understand the class. Please, next class, tell me what's the problem. We can fix it. Have a nice day. Thank you so much. Bye bye.
Bye.